So let's just introduce in this part antibacterials and some general principles about antibacterials, and then we'll go on to the various classes of drugs that belong to the antibacterials. We're not here to talk about microbiology, but we do need to briefly mention in the bacterial line, one major way of classifying bacteria is whether those bacteria are gram-negative or gram-positive. Now, the Gram stain is a stain that was developed by a man named Gram, hence its name, and it's basically a way of distinguishing between these two major groups of bacteria based on whether the bacteria readily take up the Gram stain or whether they don't. This diagram shows the major differences between bacteria which are designated as gram-positive because they do take up the Gram stain, and gram-negative because they don't take up the stain. Now, if they do take up the stain, they take on purpley colour because that's the colour of the stain. This diagram here is showing you why the gram-positive bacteria take up the stain so readily. It's because gram-positive bacteria have this cell wall which has multiple layers of peptidoglycan, like a peptides, which form this thick cell wall, and it's this component of the bacteria that absorbs the stain very readily. Compare that in the gram-positive bacteria with the gram-negative bacteria. You can see, although their cell wall is there, the peptidoglycan layer is there, it's much, much thinner, and it's hidden underneath this outer membrane. So the part that takes up the Gram stain in a gram-positive bacteria is literally on the outside, and it's very thick. So you get very marked staining compared to the gram-negatives, where that same component of the cell wall is, one, much thinner, and two, tucked behind an outer cell membrane. So these bits here where you can see the bilipid layer here are cell membranes, rather similar to the cell membranes that our own cells have, but in the gram-positive bacteria there's an inner cell membrane and then an outer cell wall, which is the one which takes up the Gram stain, and in the gram-negative bacteria there's an inner cell membrane, the cell wall which is much thinner, and then an outer cell membrane. And also noted on here that the outer cell membrane contains the lipid layer, but lipopolysaccharides in this lipid layer, which are quite strongly antigenic in us, and so that with gram-negative bacteria, should the membrane be broken down to release those lipopolysaccharides, they can cause serious illness in humans. The other term for those lipopolysaccharides is endotoxin, which you may have heard. So gram-negative bacteria don't take up the Gram stain, and they tend to have endotoxin, lipopolysaccharide, in their outer cell membrane. Gram-positive bacteria do take up the Gram stain. They don't have the outer membrane with this lipopolysaccharide in, so they do not have endotoxin. Gram-positive bacteria are perfectly capable of producing toxins which have damaging effects, but they are exotoxins which are released from the bacteria and go free to cause problems. So those are some major differences between the gram-negative and gram-positive bacteria. Some of the worst disease-causing bacteria belong to the gram-negative group, but there are disease-causing bacteria belonging to both groups, as you'll see when we come to look at the antibacterials. And some of the antibacterials are more effective against gram-positive and some against gram-negative. Okay, so along with the principle of chemotherapy then is this targeting of bits and pieces, chemical targets in a bacterium that are not present in a human cell, and therefore the selective toxicity. And so antibacterial research has been around, let's try and find out what processes go on in bacteria that are different from ours, what structures are there that are different from our cellular structures, so that we can target those in order to get selective toxicity against certain bacteria. Now, of course, there are lots and lots of different types of bacteria with slightly different characteristics, so even getting targets for antibacterials is quite a big ask, let alone other types of microbes. But let's have a look at the major drug targets that have been used to try and nobble bacteria with selective toxicity. First of all, there's the cell wall. Particularly in those gram-positive bacteria where they've got a really thick cell wall, there it is there. They have to make that cell wall, and it's quite a complicated process because it's almost like brickwork. It's like a very protective, very thick wall, just like security walls that we would have with interlocking bricks, which have to be biochemically connected to one another. So the making of that wall and the repair and the maintenance of that wall is quite an important process in particularly gram-positive bacteria, but gram-negative bacteria also have a cell wall. It's just not quite as thick. Our cells do not have cell walls. We just have cell membranes. Our cells are very delicate little things by comparison. They don't have a big, thick, protective brick wall around them, and so that is an area where you would hope, if you attack the cell wall, that would leave your cells pretty much untouched. So that's a big area. What else have we got? Well, there's the area of protein synthesis. Bacteria use ribosomes to make protein from the genetic codes contained in their DNA. They also have RNA, but the composition of their ribosomes is not exactly the same. The way that they produce the proteins, while it is the same system, there are differences between the ribosomes that we use to produce proteins and the ribosomes that bacteria have to produce proteins. So that's another point of difference that can be targeted and is targeted by antibacterials. We can also target the cell membrane by punching holes in the cell membrane. Now here we're getting a bit closer to home because of course our cells have cell membranes, but the exact composition of those lipids is not exactly the same in eukaryotic cells, in our cells, as in prokaryotic cells like bacteria. So there is some scope for exploiting the slight differences in the cell membrane structure and punching holes in the cell membranes of bacteria. It's less promising perhaps than other areas, but it is one point of difference. Another biochemical process that goes on is the synthesis of folic acid or folate. Now both in bacteria and in mammalian cells, in our own cells, folic acid is very important metabolically. It's required for the production of RNA and DNA. So your cells cannot divide properly unless there's a good supply of folic acid. So folate synthesis within the bacteria, if that can be targeted, then you can prevent the synthesis of DNA and RNA and therefore the division of bacteria. And one more area is DNA or an RNA synthesis. Again, if you can directly affect that, you can stop the copying of DNA, the synthesis of RNA within bacteria without affecting your own ability to produce DNA or RNA, then again you can stop the cell from dividing. So these 2 points of attack are really kind of on the same target but slightly different points of the target. Okay, so those are the areas that major antibacterials that we currently have in our arsenal are attacking. And there is an urgent need now for us to find new targets, because the drugs that act at these targets, by and large, with a couple of exceptions, have now got some serious resistance problems in that the bacteria, not surprisingly, have not just sat there and taken it from us. When we've produced all these antimicrobials, they divide very rapidly and they have evolved mechanisms to evade the antimicrobial actions of a lot of the drugs that have been specifically targeted against these points. And so there's a need for us to find more if they exist. So do our antibacterials kill bacteria, or do they just stop them from dividing? Well, some of them do one and some of them do the other, and some of them do both. This cartoon's rather nice because it shows it in a nice visual way. The ones that simply prevent the bacteria from reproducing or dividing are known as bacteriostatic. They obviously don't remove the bacteria if they don't kill them, but they allow the numbers to remain relatively low, give your immune system a much better chance of getting on top of and getting rid of the infection. And I've listed some of the drug classes that are considered to be bacteriostatic here, and we're going to talk about these drug classes, so it will mean more. So a whole load of antibacterial agents are bacteriostatics. On the other hand, you have some drugs which are considered bactericidal. Here's examples of the drug classes that act in that way. Now they actually kill bacteria, and so you would expect that if you had the dose right and if the antibacterial was effective, that it would actually completely clear the infection. Now we're showing this as a very clear distinction between two. There are a number of drugs which can be bacteriostatic at some concentrations but then bactericidal at higher concentrations, and so there's a fair bit of crossover between these two. And there are also drugs that are bacteriostatic against some types of bacteria, but bactericidal against other types of bacteria. So it's not as clean a distinction as this slide suggests it is, so bear that in mind. You can see probably that when it comes to the problem of resistance of bacteria to antimicrobial drugs, that you might think that it would be better in that case to have bactericidal drugs, because if you don't leave any bacteria alive, then there aren't any bacteria who've survived treatment by a particular antimicrobial to produce a new generation of resistant bugs. How important that is probably remains to be seen. There are some people that would say it's not really significant whether you use a bacteriostatic or bactericidal, and there are others who say now that we're dealing with a lot of resistance that it is more important. You'll see these terms coming up as we go through, and essentially that is all that we need to know for now, is what they mean. Here's a graph which illustrates it quite nicely. If you were to measure the numbers of bacteria and plot on a logarithmic scale those numbers, because a bacteria that's multiplying can multiply exponentially and you'll get to huge numbers of bacteria very rapidly. So we've put them on a log scale there against time. You can see that the red dotted line is bacteria that are being treated at this point with an antimicrobial that is bactericidal. Now what you'd expect to see is that a little while after you've initiated treatment, assuming that the bacteria are susceptible to that antimicrobial, that the numbers of bacteria would fall, and if you've got the dose right and the length of treatment correct, then you'd expect, as by the time the course is finished and you've stopped taking the antimicrobial, that you've got very, very low numbers of bacteria present, and perhaps even none. Whereas with the bacteriostatic drug, if you start the treatment at the same point as you started with the bactericidal, the same number of bacteria, then what would happen is the bacteriostatic wouldn't actually reduce the number of bacteria from that level, but it would prevent the numbers going up too much, at all, possibly. And therefore, when you stop taking that course, then you would expect the numbers to start increasing again, because you haven't actually killed the bacteria, but you've just stopped them from reproducing. Now, of course, this graph does not take into account the effect of the immune system and your antibacterial immune cells that would be getting on top of the bacteria as well. But this is what would happen if you just took the immune system out of the picture, that if once you removed a bacteriostatic drug, then you would have those bacteria still there, which would then start to reproduce again.